This is a slide on what's happened to sequencing since the Human Genome Project in the first decade and a half of this century. And all I want you to see from this is logarithmic. The cost of sequencing is diving, and the capacity of one sequencer is shooting up. I'll show you a little detail. This is Solexa from Illumina. So the idea, and in a way, this still has Sanger's use of DNA polymerase, but it makes it parallel by factors of millions. So what happens, this is our unknown DNA here, and we get attached known pieces to the ends of these unknown things, and they started out to be about 50 base pairs, they're now bigger. And then separate this guy, get this guy into single strands, and with incredibly cleverness, they get these adapters to attach on a surface. This surface is covered with sticky places for these adapters, and the pieces land here and there. And then there's a really intricate, if you want to learn how smart people in biotech are, look in detail at this process, but I can outline it. They managed to get this piece to bind again. They add the DNA here, and then get two where there was one, and you amplify locally. You get amplified this unknown piece of DNA to be a cluster of stalks at different unknown places on this slide. Think about the size of a computer chip. And then, let's see, let me move ahead. So then this I like this picture. So here's the DNA polymerase going along, and at each step, a base is added, and the bases are still labeled with these fluorescent dyes. So if you get a picture, I don't know what, green, blue, yellow, and red probably. I'm slightly colorblind, so it doesn't depend on me being able to see these, but this spot would mean that a red base was added to the unknown sequence at this location. So you get a whole bunch of unknown pieces that are located variously on this slide. So again, increasing making Sanger sequencing parallel by factors of millions. If there's one slide I'd like you to read, this can easily be found on the web. One slide I'd like you to take home with you it would be this slide. So on this axis, oops, well, that worked while it worked. There it goes again. I'll just try. So this is the cost of 1 million bases of DNA sequencing starting the year 2001, where it was a little under $10,000, which sounded cheap to me at the time. Here's Moore's law, and it's the version of Moore's law that doubles every year and a half. So that straight line is Moore's law, and here's the cost of sequencing those guys. And it's sort of leveled off now at something like 8 cents a million base pairs. So we're in the world of going from spending $3 billion to sequence a human genome to slightly less than $1,000 sequencing a human genome. And I claim there's no question there's another factor of 10 in there. So we will all, or you all who are slightly younger than I am, will be having your genetic sequence when you see the doctor. Pretty amazing. Now, learning what all this means is another much more confusing topic. But if we were going about it in the same way as we did before, this just will not go anywhere. It's just too computationally intensive to continue on the Roger Staden approach.